Assalamualaikum dear students, welcome to MLT DEC0DE. Today is the molecular biology playlist and the MCQs video. So let's start. The first MCQ is in 1869, John Frederick Michener discovered DNA, RNA, blotting techniques or all of these. Right is DNA. The next MCQ is which of the following process does not occur in prokaryotes? Translation, transcription, splicing, and replication. The right answer is splicing. The next is which enzyme is used in unwinding of DNA? Topoisomerase, helicase, RNA polymerase, or DNA polymerase. The right is helicase. The next MCQ is which enzyme is used to join nicks in DNA strand? DNA ligase, isomerase, glycosidase, DNA polymerase. The right answer is DNA ligase. The next MCQ is the messenger RNA codon of valine is AUG, TTG, GUC, UGG. The right is GUC. The next is DNA synthesis is measured by estimation of incorporation of a radiolabeled cytosine, uracil, thymine, adenine. The right answer is thymine. The next is in PCR, the temperature at which DNA synthesis takes place is 94 degrees Celsius, 74 degrees Celsius, 104 degree or 54 degree. Right is 74 degrees Celsius. The next is automated sequencing is defined as chain terminating sequence, real-time fluorescence sequence, radiolabel sequence, or none. Right is real-time fluorescence sequence. The next is automated sequencing uses radiolabeling, fluorescent tagging, enzymatic process, or none. Right answer is fluorescent tagging. The next is how many primers are used in the reverse transcription amplification? 4, 6, 2, or none. The right answer is 2 primers. The next is from which organism the enzyme reverse transcriptase is used? Bacteria, parasite, viruses, fungi. The right answer is viruses. Which of the following not required for DNA sequencing? PCR, cloning, gel electrophoresis, restriction digestion. The right answer is cloning. DEC method is also called polymerase chain reaction, auto-sequencing, Sanger enzymatic method, all of these. Sanger enzymatic method is the right answer. The hairpin loop of RNA in termination step consists of nucleotide in abundance AT, GC, UA, all of these. Right is G or C. The next is the raw protein have how many subunits? 8 subunit, 2 subunit, 4 subunit, 12 subunits. The right is 6 subunits. The next is which base undergoes spontaneous damage under physiological condition which leads to generation of uracil? Adenine, thymine, cytosine, histidine. Right is cytosine. The next is what is the product formed after oxidation of guanine 2,7-dihydroate guanosine, 7,9-monohydrate guanosine, 7,8-dihydroate oxoguanine, or all of these? The right answer for this is 7,8-dihydroate oxoguanine. Next is 5-bromouracil, a anti-cancer drug, is analog of thiamine, guanine, uracil, cytosine. The right answer is thiamine. Next MCQ is cholesterogenic agents are those which break RNA, which break DNA, which break cell, which break polymerase. The right answer is which break DNA. Next is bleomycin is anti-cancer drug which break DNA, peptidoglycan, reverse transcriptase, or cell wall. The right answer is DNA. The next is polymerase used for PCR is extracted from Staphylococcus, E. coli, Thermus, Aquaticus, Homo sapiens. The right answer is Thermus aquaticus. Next is amplification is carried out by which enzyme from Thermus aquaticus? DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, or all of these? Right is DNA polymerase 1. Next is what is probe? Purified DNA, fragmented RNA, purified or synthesized single-stranded DNA, purified or synthesized single-stranded RNA. The right answer is purified or synthesized single-stranded DNA. Next is northern blotting is performed by DNA detection, quantification of protein, quantification of RNA, quantification of viruses. The right answer is quantification of RNA. The next is the host bacterium take up plasmid in the presence of divalent anion, monovalent cation, monovalent anion, divalent cation. The right answer for this is divalent cation. Next is the largest molecule in our body is DNA, protein, chromosomes or cell. The right is DNA. Next is portion of DNA that does not code for protein are called exon, intron, codon or none. The right is introns. The next is which of the following biomolecules have repair activity after damage? DNA, RNA and protein, DNA and RNA, RNA only, DNA only. The right for this is DNA only. The next is which of the following is bypass repair system? BER, SER, SOS or none. The right is SOS. The next is the distortion of DNA helix due to pyrimidine dimer formation is Nike, Knick, single strand break or all of these. The right answer is Knick. The next is operon or unit of coordinate change expression only found in eukaryotes, prokaryotes, both or none. The right is prokaryotes. The next is catabolic activity protein only active when glucose level is high, moderate, low or all of these. The right is low glucose. The next is histones are protein which are highly alkaline, acidic, neutral or none. The right is alkaline. The next is the Ligase gene encode enzyme that split lactose into dimer, monomer, pentamer or none. The right answer is monomer. The next is acetylation and methylation of histones make it highly negative, positive, neutral or none. The right is negative. The next is eucoatination is the process of addition of lipids, proteins, carbohydrates, all of these. The right is proteins. The next is if there are 30% adenine present, then what percentage of cytosine must be? 30%, 20%, 60% or none. The right is 20%. Next is RNA primer is synthesized by the enzyme polymerase, primase, helicase or topoisomerase. The right is primase. Next is which of the following is stop codon? UAA, UAG, UGA or all of these. Right is all of these. Next is rifamycin, an antibiotic block initiation of RNA chain by binding to DNA polymerase, RNA polymerase, DNA gyrase or all of these. Right is RNA polymerase.